Part of the quantum research. 01. Christian Daniel Assoun. The third catenary of DNA or RNA. The UV radiation, X, Y, gamma, inside the biologic material. Introduction. Quantum realities and paradoxes, apparent. 2 records, tables, will lead our study applied to radiation in the cell materia. Natural elements in the universe. The DNA. 1. The natural elements in the universe. The complexion of the 3 fundamental disciplines. Astrophysics. The atomic physics. The physics of particles. Has caused great changes in our beliefs on the matter, ion energy, and memory governing the establishment including ours. These disciplines are still in childhood if one considers the probable age of our universe, 13 billion years, and the biological kingdoms, which man, who on Earth would not land more age than 3 million years. Our basic sciences are not older than a few centuries. We must feed much hope if man of this earth has happened to hundreds of thousands of years scientific development in maintaining the speed of progression of knowledge, offset by wisdom. We can also caress and hope it exists in our world civilizations that have achieved what we want to achieve since the eve of humanity. But the goal of our work is modest to ask the first scientific basis and experimental called this discipline. Quantum medicine. Everything is arranged by the number announced. Pythagoras, the Mendeleev table, page 7, is a cosmological and biological demonstration. For those curious about the natural and applied sciences, it is good to know that the first ancient physicists and chemists, Annie Besant and Leadbeater CHW 1920, in a magnificent work, entitled Occult Chemistry Joining the Field of Atomic Physics. Recently 2014 modern physicists discovered that nuclei of free atoms were organized in particular shapes and seems to be identically at those described in the book of Annie Besant and Leadbeater CHW 1920 considered as esoteric approach by modern physicists. We can only evaluate the atomic and quantum resonance between matter and our brain who is working as quantum machine. This is the main logical approach, because how we could interpret and listen quantum chemistry or physics if our brain was not organized as performing memory and tool for our future evolution. Somewhere in our brain and neuronal tissues are located center more adapted to quantum physics or quantum mechanics, and by the resonance we can touch these registers and extract by MHD magneto-hydrodynamic process the solutions of problems. This quantum view of neuronal matter do not pull out the existence of something great in the universe, as the contrary. Past scientists working with esoteric researchers represented initially located and read, meditation, the natural elements and their approximate atomic masses on a Mendeleev table or even double or triple helix, and Violent announced the discovery that a few decades later biologists and biophysicists, Crick and Watson, 1953, were discovered. The esoteric approach was not so crazy because, in fact decades after all atomic and biologic representation were demonstrated by modern physics and biologic tools. 2. DNA and RNA. The double helix of our DNA equals chemical material basis of our genetic. For the moment, no experimental theory elucidates the subtle mechanisms of differentiation cell lines, mitosis, and the self-repair of DNA. Biologists or geneticists can intervene by changing the conditions, catalysis the promoter's reactions, DNA replications, but no one can now give an electromagnetic information is a substance or material DNA nucleotide to him perform a specific biological or biochemical mission. Only the memory transfer bases. AGTC catenary base associated with specialized enzymes PGR, polymerase chain reaction, may again recreate the original medium in a neutral medium containing no DNA. These experiments is a memorial transfer, instructions, to give developers a PMP so that these promoters reconstruct DNA from a memorial picture. It will be more than a memorial, a teleportation area of DNA transfer. Colleague prestigious work of Ben Venist, echoed by other researchers, including Prof. Montaigne have demonstrated the natural propensity of the genetic material has to remember his love and genetic problems via a curious substrate Rha. The UAE is the first quantum liquid, its state liquid is present then its gaseous state should be. Water is a matrix in the form of quantum monomers or polymers in our cells. The development work on the electromagnetic. The water beyond the scope of this limited submission. Already in 1986 they laid the foundations of the elucidation of viral infection by HIV concept electromagnetic in a private communication, the mechanisms of passages of HIV in membranes. Physicists can create laboratory plasmas to better understand their mechanisms macroscopic and microscopic course. It is found that the plasmas dotes appear of intelligence surprising. Creation of laboratory plasmas, Professor C. Diasoun, Glycan Group. Argon Plasma, Glycan Lab. Imagine inductor's high-frequency field, black spiral copper, representing the double DNA helix and central plasma created in our laboratory, argon or hydrogen or others. This configuration reminds our DNA laboratory and center ionized plasma, HAR, other gases plasmagons. In fact, our biology is naturally plasma, it combines the necessary four elements thuator and fire, energy, earth, minerals, and air, oxygen and other rare gases, and five registered element, the plasma, but also music with an impressive procession of wave packets electromagnetic, called observable within the meaning of the formalism of quantum mechanics. The conductor is the basic memory, DNA, and its codes, exon and intron, that genetic and biological data work are just and perfect, or almost. 3. Scientific basis and experimental medicine quantum. 1. Phenomenon in quantum DNA, the triple helix, 5 to 7 other configurations helical possible in study. We lay on our side the presence of a triazine-stranded DNA or catenary, the center of the biconventional catenary shape of nucleotide bases, AGTC, U, our research in 1982 to 1986. This third strand or catenary shape is a plasma of hydrogen, H, in NH oscillation. Transitions. This atomic hydrogen plasma are the source of a UV, ultraviolet intense, radiation resonance frequencies are the same as those from the cosmogonic material containing hydrogen atoms in the ionized form at high temperatures, in K000, so 000K. TT equals plus 273.14. At these high temperatures the difference in gradient of 273.14 degrees Celsius, between Kelvin, and has no C importance, in some cases I00000K, I. We can write 30,000 3000 degrees degree C, to be simple. These temperatures are real but their life, atomic transition, lasts only a few millionths or billionths of a second, about 10 registered s. We are talking about plasma oscillation due to the extreme short-lived transition. Each millionth or billionth of a second depending on the temperature, of ionization states I, 2, 3, oft hydrogen, our DNA emits UV radiation but also or even XY, gamma. This radiation has been described as ultra-low by researchers Pop and Lee who find experimentally. Pop and Lee do not pose experimental hypothesis state plasma hydrogen, which seems to be the only one able to realize the quantum state of our system biological, cDNA or RNA registry. 2. Quantum phenomenon, located in the membranes, nucleus and cell, according to our research from 1982 to 1986 to date. Our cell can be considered an open quantum system and closes whenever packets of energy entering or, instead, out of a cell. The quantum reasoning is applicable only to cells having a core provided with its DNA. For the moment the elucidation of quantum phenomena in nucleated cells is not clear even if the membranes of these cells are the site of important energy phenomena. The membranes of nucleated cells have very small dimensions of about 5 nanometers, 5.10 meters or 5.10 centimeters. These mechanisms have been elucidated in a private communication called Reason Use Lanthanide, rare earth in uncomplexed in cellular dietary form. Elucidation of quantum phenomena of synergy and coherence in biological systems. DNA mitochondrial. Reading coding Hermitian. Gas intermembrane proton. The genetics. Quantum, C.D. Sound, March 1985. It is shown by calculation that the ions pass through the membrane in performing their path initiatory few millionths of a second was the actual speeds that can reach 14 km per second 280 km per second DDP membrane, potential difference, and field E equals U/D for ions can vary 100,000 volts. 200,000 volts. This accelerator is a field to origin the velocity ions, obviously. These ions in turn collide with other ions and the shock is exothermic. 2 causes ionized states throughout the ionic material of cells. Ensemble the table natural elements is present in our cells, see page 8. The actinides, in principle, are not present in our biological material, except for uranium and thorium. Lanthanides or rare earths are essential in protein synthesis. This statistical and quantum-based approach lectures known of mechanics quantum whose best works are those basic. Professor Albert Messiah, our master, has almost everyone. Messiah delivers a significant portion of the elucidations of mechanisms to apply both to our cosmogony what our genetic material DNA, RNA that delivers only 5% of its secrets with the parties coding exon, 50,000 proteins. Our work is currently focused on the intronic parts which represent 95% of our DNA and classified unjustly silent or even useless. Non-coding parts are referred to as introns, an easy dogma, which has already undergone numerous violations and fall, like other doctrines drawn up by necessity. The intronic part is at the origin of more than 570,000 transitional metastable proteins and fractals which are DNA. To organize a little better his or her longevity immunoresistance, could build its new structures 8 of new tetravalent elements such as germanium or silicon, properties optoquantics reverses. This work, New Frontier of DNA, of nucleophilic substitution on carbon will take decades research in specialized laboratories in quantum science with banned budgets for time due to financial crises. 4. Our planet in the universe pre-Glycan dimensions unrealistic. The cosmic level it is assumed that even if nature abhors a vacuum, our known universes form 99% of subjects in all genres I-state plasma, also denominated fourth state of matter. One universe? Some researchers mention flashing 63 plus 1 universes. Register memory of DNA, 5. The center of the DNA is registry memory and presents the plasma hydrogens. Harmonic. OHHNHO, 2x3, NHOH, 2x2. S equals synergy. C equals consistency. DNA/N equals part statistical mixtures. V equals volume hemioteque registry, containing quantified information. M equals memory. 6. Detailed information on the foundations of quantum medicine. Prior to the fourth state of matter, plasma state, exist other 3 states, namely solid state, the state liquid, the gas state. 99% in a plasma state, astrophysicists include 75% dark matter would be a plasma crystallizes in another repository. The first observations defy researchers but do not be discouraged. The remaining 1% consisting of condensed matter hot or cold formed by natural elements known or unknown. Temperature gradients are different from one place to another in our universe, it oscillates between 0 is -273 degrees Celsius and 14 million Kelvin for stars of all kinds. Astrophysical observations, remote or CMB, and gravitational tell us about the presence of the trailing edges of the universe, black masses of proto-matter or the form crystallized. One must say that the color is not really color, blue, green, yellow, red, but statistically just as he seems, for the properties of quarks, which are statistical or chromatic colors, quantum particles and subparticles. Some specialized in plasma physics and condensed matter physicists. There exists the trailing edges of the universe, which look like a fried egg, pressures so enormous crystals would form, and consisting of time, these crystals should have different colors. According to a particular formalism, there could be 64 forms of time, like 64 colors statistics. This has been developed in part in the work on quantum medicine intronic CDA sound. And more original, time would tell Morphon's form of memory structures, and time would have a weight according to the calculations. 2 exists a root color and color diffraction or fractals according to the standard adopted. For example, in the most known examples of atomic spectroscopy, when the atoms are excited according to the process of obtaining a plasma radiation, each atom can emit in the spectral range of thousands known electroquantum transitions pass by when the ground state, stable, to the excited state, quantum, returning to its ground state every 10 seconds 10 dry, Fig 3. Billions and billions of light quanta, HV, are emitted and detected by spectrometers and spectrographs. These thousands of atomic spectral lines represent the true identity of a stable or radioactive atom. It can be argued that this distribution is gene. E equals HV equals 6.62510 erg.s, Planck's constant. V equals is the frequency of the associated transition. There are well gives homogene states meet in both large geometric dimensions of our universe in atoms and in our DNA. There is a geometric harmonic I order 9 either 10 or 10 registered 30 with C equals 0.10 cms1 registered speed of light, and the DNA registered with 3.10 bp, base pairs. Everything seems to arrange by the number 1. Posing an original scaling, Ton could mention about our basic genetic material, DNA-tRNA, that 5% the layout coding bases, exons, AGTC, U, and 95% of bases do not encode introns. So a universe is subjected to a morphogenic distribution of this order, that is to say that there would be 5% of known material, or almost, and 95% of unknown material, or almost, that dark matter and plasma. In this place study it's good to note that the presence of 75% or 95% of material has stall plasmas, some stranger than others, proves how necessary and sufficient that physicists and biophysicists interested in this fourth state of matter or plasma state and seek his presence in our biological life and gives its physical manifestations in our biological material. Preliminary conclusion. Only multi-complex interaction between basic disciplines will create a new formalism, as when classical physical mechanics needed to resort to quantum mechanics can account and create new experimental concepts that understand the reality of matter and present life in our universe. 7. Important information on music DBS atoms. Quantum transitions, atomic rays, UV radiation, exist by the thousands for a single atom. These atomic spectra exist in our cells, just like in the star training. References, works, publications and international conferences. Agaverdiyev, A.Sh, B.N. Tarasov, Biophysics 10, 387, 1965. Agaverdiyev, A.Sh, Y.A.Ye. Doskok, B.N. Tarasov, Biophysics 10, 920, 1965. Artemev B.V., A.S. Galdabin, L.N. Guskov, Biophysics 11, 1278, 1967. Barenboim, G.M., A.N. Daminsky, K.K. Turovarov, Luminescence of Biopolymers and Cells. Plenum Press. N.Y 1969, p.124. Barnathi, J. Biological Effects of Magnetic Fields, Volume I Plenum Press, N.Y 1964. Blaka, V.V., G.V. Kosova, A.D. Sizov, V.A. Fedin, U.P. Kozlov, O.R. Coles, B.N. Tarasov, Biophysics 1? 921. 1968. Cali, Lu, Facchini, Nuovo Cemento 12150, 1954. Cali, L. U. Facchini, G. Guidati, R. Dugnani Lonati, M. Orsnigo, A. Samariva, Experientia 11, 479, 1955. Dorman, Ya.G. Biophysics 7, 733, 1962. Doskok, Ya.Yev, AP. Yakovlev, B.N. Tarasov, Biophysics. 596, 1969. Gurvik, A.G., S. Grabsch, S. Alkind, in, Arch F. Ent.Mech 100, 11, 1923. Gurvik, A.G., L.D. Gurovich, Die Mitogenetisk Strahlen, Jena 1959. Hollander, A. Court. Reverend Bio. 246, 1960. Koenig, H.L. Unsichtbarumwelt, Moosverl, Munchen 1975. Konak, S.V., T.I. Liskova, G.D. Nisenbaum, Biophysics. 410, 1966. Mamedov, T.G., G.A. Popov, S.V. Konak, Biophysics 14. 1102, 1969. Palmer, J. Nature 1. 1061, 1963. Perligrin, B.V., B.N. Tarasov, Biophysics 11, 616, 1966. Pop, F.A. In, F.A. Pop, G. Becker, H.L. Koenig, W. Peshka, eds. Electromagnetic Bioinformation, Urban. And Schwarzenberg, Munchen, Baltimore 1979. Pop, F.A., H. Klima, H.G. Schmidt, Biophoton Physics 1, 3, 23, 1979. Pop, F.A. Umsho 7, 235, 1979. Priesman, A.S. Electromagnetic Fields and Life, Plenum Press, NY 1964. Ruth, B. F.A. Pop, Z. Naturforsche. 31c, 741, 1976. Ruth, B. Dissertation, Marburg 1976. Ruth, B. In, F.A. Pop, G. Becker, H.L. Koenig, W. Peshka, eds. Electromagnetic Bioinformation, Urban and Schwarzenberg, Munchen, Baltimore 1979. Shiyaktina, L.S., A.A. Gurvik, Biophysics 17,1146, 1972. Zanpurji, A. Interaction of the non-ionizing electromagnetic radiation with living systems. Versailles 1979. Veslovsky, V.A., Ye.N. Sikimova, V.N. Tarasov, Biophysics 8147, 1963. Yafarava, I.O., V.A. Veslovsky, Biophysics 14, 390, 1966. P.C. Newell, Endeavor New.Serl, 63, 1977. B. Worcester, P. Penn, G. Tyen and J. T. Bonner, PROC. National Academic Sci. USA 7, 795, 1976. J. F. Whitfield, J. P. McMahons, R. H. Reichsen et al. In vitro 12,1, 1976. A.J. Clark, Mode of Action of Drugs on Cells. Arnold, London 1933. H.N. Schultz-Salche, Dissertation, FB Human Medicine, Marburg 1978. WDM. Patent, proc.roy.socserb. 1. 21, 1961. I. Prigozhin, Nobelvortrag, Deutsche Übersetzung. Eng. Chemistry. 704, 1978. H. Hacken, Synergetics. Springer, Berlin Heidelberg N. Y. 1977. B. Hess, A. Boyt-Lux, Hoppe Sealers Z. Physiology Chem. 349, 1567, 1968. L. Renzing, G. Cornelius, Bile.RDSCH. 18, 197, 1980. G. Schaefer, Lieboesen Modell. In Lytheman? Westermann, Braunschweig 1977. F.A. Pop, in, F.A. Pop, G. Becker, H.L. Koenig, W. Peschka, edits. Electromagnetic Bioinformation. Urban and Schwarzenberg. Munchen, Baltimore 1979. H. Jenny, Zymative. Baller UND Berlig Sandstelt. Basilius Brasa AG, Basel 1974. Th. Schwenk, Das Sensible Chaos. Stromens Formenschaffen in Wasser UND Luft. Verlag Phrase. Geisleben 1976. H. Fröhlich, INTJ. Quantum Chemistry 2. 641, 1968. F.A. Pop, Physical.med.u.rad. 334, 1980. H. Hacken, Z. Physics. M. 96, 1964. H. Guntherud, Physik in unserer Zeit. 74, 1978. G. Grölich, Erfahrungscheckend, F. 7, 575, 1980. F.A. Pop, H. Kayama, H. G. Schmidt, Biophoton-Physics 1, 49, 1980, Erhaldlich, Biomet Verlag, Out Dem. Rosenberg 2, D5307 Wattbart. F.A. Pop, Umshow 8, 235, 1979. F.A. Pop, in, proc.lnt.symp. On Analytical Applications of Bioluminescence and Chemiluminescence. Brussels 1978. E. Scram, P. Stanley eds. State Printing and Publishing Incorporated, Westlake Village 1979, pages 601. F.A. Pop, in, Molecular Base of Malignancy. Selected papers from an intern.symp. E. Deutsch, K. Moser. H. Rayner, and A. Stacher eds, theme, Stuttgart 1976, pages 47. F.A. Pop, Biophotonen. Schriftenreihe, Krebs Geschichten 6, Verlag für Medizin 3 V. Fischer, VFM. Heidelberg 1976. F.A. Pop, V. Strauss, Soken Krebs Entstehen. Fischer TB 6800, Frankfurt 1979. W. Nagy, F.A. Pop, in Vorb. F.A. Pop, B. Ruth, W. Barr et al. Collective Phenomena, in Press. F.A. Pop, H. Kayama, B. Ruth, in, Bericht von der XXVI. Tagung über die Zytoplasmatische Therapie vom 3-5. OKT. 1980 in Stuttgart, Ank Verlag, Stuttgart. F.A. Pop, Laser UND Electro-Optic. 28, 1980. W.R. Bauer, F.H.C. Crick, J.H. White, Spectrum der Wiss. 25, 1980. M. Ratimeter, W. Nagy, F.A. Pop, Nature Wissenschaftern. M. Eckhorn, Bio RDSCH 18, 241, 1980. J. Viscar Math. Bio. 29, 1976. V. P. Kaznachev, S. Shurin, L. Mikhailova, Naturug. RDSCH. 444, 1973. V. P. Kaznachev ETAL, Bulletin Experimentarnoi Biologie I Medicini 87. 468, 1979. H. G. Schmidt, F. A. Pop, in Vorb. P.S. Callahan, Tuning into Nature. The Devonadair Comp, Old Greenwich, Connecticut 1975. P.S. Callahan, Physio. Chem. And Physics 1? 31, 1980. E. Yance, C. H. Waddington, edits Evolution and Consciousness. Addison Wesley P.C., Reading. Massachusetts 1976. P. T. Saunders, M. W. Ho, J. The. Bile. 6. 375, 1976. W. Nagy, PLSYST.EVOL, Supple.2, 3, 1979. J. Wiccan, J. The. Bile. 77, 349, 1979. A. St. György, Introduction to Submolecular Biology, Academic Press, New York, 1960. A. St. György, Towards a New Biochemistry, Science 93, 609, 1941. B. Pullman, A. Pullman, Quantum Biochemistry, Wiley Interscience, 1963. R. A. Pething, A. St. György, Electronic Properties of Casein Methylglyoxyl Complex. PROC.NAT academic.sci.U.S.A, 74226-8, 1977. D. Ely, Semiconducting Biological Polymers P.259-294, in Organic Semiconducting Polymers. Ed. E. Keaton Marcel Decker, New York. Rosenberg, Electrical Conductivity of Proteins, Nature 193, 354, 1952. D. Ely, D. I. Spivey, Semiconductivity of Organic Substances, Part 9, Nucleic Acids in Dry State. Transaction Farad. SOC 58,411-5.1962. J. Ladik, The Energy Band Structure and Conduction Properties of DNALNT. J. Quantity Chemistry, Quantity Bile. SYMP. No. 1,65-69, 1974. B. Rosenberg, J. F. Camisoli, Photo and Semiconduction in Crystalline Chlorophyll. A and B. J. Physical. Chemistry 35. 982, 991, 1961. R. J. Cherry, D. Chapman, Effect of Gases on Electrical Conductivity of Beta-carotene, Nature, 215, 956-957. 1967. G. Caserta, T. Cervini, Piezoelectric Theory of Enzymatic Catalysis as Inferred from Electromechanical Principles of Bioenergetics, PROC.NATL. Academic.sicu.s.a, 71, 4421-4424, 1974. F.W. Cope, A Review of the Applications of Solid-State Physics Concepts to Biological Systems, J. Bile. Physics. 3, 1-41, 1975. C.A.L. Bassett, Biological Significance of Piezoelectricity, Calcif. Tis.res1. 2s2-212, 1968. E. Foucault. Piezoelectric Properties of Organic Polymers and New York Academic. Sci. 238, 7-25,1974. H. Athens. Permanent longitudinal electric polarization and pyroelectric behavior of collage news. Structures and nervous tissue in man and other vertebrates, Nature, 228, 838-34,1970. A. Zentburgi, Charge Transfer and Cellular Activity p. 192 to 193 in Theoretical Physics and Biology, 4.1 STINTCONF. On Theoretical Physics and Biology, Versailles, 26 to 30 June 1967. De. M. Marik, North Holland, Amsterdam, London, 1969. L. Bozicki, G. Kisle, T. A. Hoffman, J. Loddick, Effect of Electrostatic Fields on Cell Mitosis, Nature, 199,1306. 1963. W. Sedlak, A. B. Koft, Electromagnetic Theory of Life, in Polish, Cosmos A, 18,164-174, 1969. G. Lochowski, The Secret of Life. Cosmic Rays and Radiations of Living Beings, trans. From French, 2 ed. True Heath Publishers Comp. Stockwell, 1951. W. Sedlak, A Model of a System Radiating the Biological Field and Electrostasis, in Polish, Cosmos A, 2,154. 159, 1967. V. M. Inyashin et al. Biological Aspects of the Glycan Effect. The Biological Plasma Concept, Alma-Ata, Kazakhstan, 1968. D. Pines, Collective Energy Losses in Solids, Reverend Mod. Physical. 28,184-198, 1956. A.G. Chinoyev, S. J. Buxbaum Solid State Plasma, Physical. Today 18, 2637, 1965. D. Ely, R. Pething, Microwave Hall Mobility Measurements on Rat Liver Mitochondria Spinach. Chloroplasts. J. Bioenergetic 2, 39 to 45.1971. M. D. Williams, J. L. Fox, De Broglie Waves in Amoeboid Mobility. Physiological Chem. Physical 8, 397-403, 1976. H. Athens. Ferroelektrisch und Piezoelektrisch Ingenschaften Bedeutsamer Staff. Nature Wissenschaften, 48, 465 to 472, 1961. J. Polanski, P. Tuzu, C. H. Sardin, Mice en Evidence de Propriétés Ferroélectriques dans Iacide. Desoxyribonucleic, DNA, Kant.Ren.Heb.Sci. 250, 3414 to 3416, 1960. J. Zonin Attempt at a Bioplasmic Approach to the Death of a Living System, PROC. Symposium on Polish Bioelectronics, Warsaw, October 22 to 23, 1977, to be published. W. Sedlak, Bioplasma, A New State of Matter p. 13 to 30, in Bioplasma PROC First Bioplasma Conference. May 9, 1973, held at the Catholic Universal of Lublin, Poland, 1976, English translation in 1978. M.R. Riviere, A. Prior, Kant.Ren.Sc. 259, Group 14, 4895-4897, 1964. R. Patrizal, A. Prior, 268, Ser. D. 1889-1892, 1969. R. Patrizal, A. Riviere, Kant.Ren 262, Ser. D.579-582. 1966. N.D. Deviatkov, Influence of Millimeter Band Electromagnetic Radiation on Biological Objects SOV. Physics. USP. 16, 568-9, 1973. VH. Inyushin, PR. Chekurov, Biostimulation by Laser Radiation and Bioplasma, Kazakh State University. Almaty 1975, English trans, available from Danish Society of Psychical Research, Copenhagen. H. Motoyama, The Ejection of Energy from the Chakra and the Meridian Points of Acupuncture, p.375. Second int. Congress on Psychotonic Research, Monte Carlo, 1975. Lingwei.Wei, AMJ. Chinese Medicine, 4, 355, 1976. V.G. Adamenko, The Electrodynamics of Life, J. Paraphysical, 4, 113, 70. T.Y. Fen, Electrical Acupuncture and Cautery Therapy, in Chinese, Hong Kong, Commerce Publ, p.113. 1972. L.C. Vincent, Rev. Path. General 677, 1956, Apricus Theoretics et Pratiques sur la Bioélectronique Vincent, Editions La Source d'Or. Marcet, 63 France. Common Documentation, Sibev. Doctorgus 8, 5963 Wenden, Ottfingen, W. Germany. Barnothy M.F/ed/ Biological Effects of Magnetic Fields, Volume 1 and 2. Plenum, New York, London 1964 and 1968. Becker R.O., Relationship of Geomagnetic Environment to Human Biology, New York State J.Med, 6. 2215-2219. 1963. Vice Chair D.E., F.F. Miller, Biological Effects of Magnetic Fields in Their Relation to Space Travel, in Bioastronautics, Schaefer K.E/ed/, Macmillan, New York 1964, pages 173-180. Vice Chair D.E., F.F. Miller, J.C. Nepton, Exposure of Man to Low-Intensity Magnetic Fields in a Coil System. NAMJ 1018, NASA R39 Naval Aerospace Medicine Institute, Pensacola 1967. Bliss VLF.H. Hefner, Circadian Activity Rhythm Influenced by Near Zero Magnetic Field. Nature, 261, 411. 412, 1976. Bressler SEW.M. Bressler, on liquid crystalline structure of biological membrane/in Rus/dope.ons. 214, 936-938, 1974. Brown F.A., Jr., Response to Pervasive Geophysical Factors and the Biological Clock Problem. Cold.spr.harb Quantity Bile, 25, 57-71, 1960. Brown F.A., Jr., Responses of the Planarian, Tuxia, and the Protozoan, Paramecium, to Very Weak Horizontal Magnetic Fields. Bio.Bull, 123-264-281, 1962. Brown F.A., Jr., Association Formation between photic and subtle geophysical stimulus patterns anew. Biological Concept, Biology Bull, 132, 311-319, 1967. Bulanda W., Electronic Conductivity of Biological Compounds and its Significance in Biology/in Polish/in Zagadnienia Biophyzyki Epoch Czeznaj, 2,15-42, 1977. Cope F.W. Biological Sensitivity to Weak Magnetic Field due to Biological Superconductive Josephson Effects. Physiol.Chemistry Phy. 173-176, 1973. Cope F.W., A Review of Solid-State Physics Concepts to Biological Systems, J.Bio.Physical, 3. 1-41, 1975. Dyke J.H. Van, M.H. Halpern. Observations on selected life processes in null magnetic fields, Anat.Rec. 151. 480,1965. Arid cf. E. Trucco, Molecular Models for the Circadian Clock. I. The Chronon Concept, J.the.bio.l. 240. 262,19. L. Derive L. Ua.Kolodov, Effect of Constant Magnetic Field on Motor Activity of Bird in Rus ZH. Obstbile 2. 224 to 229, 1964. Friedman HRO. Decker, Geomagnetic Parameters and Psychiatric Hospital Admissions. Nature, 200, 626. 629. Halpern MH. Effects of Reproducible Magnetic Fields. The Growth of Cells in Culture, NASA CR 75121. National Aeronautics and Space Administration, Washington 1966. Koenig H.L. Unsichtbare Der Mensch im Spielfeld elektromagnetisker Kraft, 2 auf I, Muenken. 1977. Labes M.M. A Possible Explanation for the Effects of Magnetic Fields on Biological Systems, Nature, 211. 968,1966. Marino A. R.O. Becker, Biological Effects of Extremely Low-Frequency Electric and Magnetic Fields. A Review. Physiol.Chemistry Physical, 2, 131 to 147, 1977. Engis D. F.M. Sulzman, J.W. Hastings, Membrane Model for the Circadian Clock. Nature, 248,116-120,1974. Engis D. V.D. Gooch, D. Mergenhagen, F. Sulzman, J.W. Hastings, Membranes and Molecules in Circadian Systems, Federal PROC. 2353 to 2357, 1976. Paulides T. Population of Biochemical Oscillators as Circadian Clocks, J. The Bio. 319-338, 1971. Persinger M.A., ELF, and VLF Electromagnet Eichel Field, Plenum, New York, London 1975. Pittman U.Z., Magnetism and Plant Growth 2. Effects on Root Growth of Cereals, Ken J. Plant Sci. 44, 283. 294, 1964. Priesman A.S. Electromagnetic Field and Life, Plenum. New York, London 1970. Writer R. Meteorobiologie und Elektrizität der Atmosphäre, Portig, Leipzig 1960. Romero Sierra C., C.J. Tanner, Biological Effects of Monensing Radiation. An Outline of Fundamental Laws. And. New York Academic. Sci. 2. 263 to 272, 1974. Iniashin, M.B., Kerlin Effect and Bioplasma Concept Alma Ata, 1968. Zahn J. Physical Plasma in Biological Solids. Physiol, Chem and Physics, 11, 501 to 506, 1979. Sedlak W., Electrostasis and Organic Evolution / in Polish Rozniki Filozofizm, 15/3/3058, 1967. Sedlak W., Physical Plasma and Laser Effects in Biological Systems / in Polish / Cosmos A, 19,143-154, 1970. Sedlak W. An Outline of Biological Magnetohydrodynamics / in Polish / Cosmos A, 2. 191-201, 1971. Sedlak W. Physical Plasma as the Base of Bioenergetics / in Polish / Rozniki Filozofizm. 20/3/125-148. 1972. Sedlak W. The Metabolism and Bioplasma Dynamics in Polish Cosmos A, 24, 261-272, 1975. W. The Evolution of Bioplasma in Polish Rozniki Filozofii 23-3 95-116, 1975. W. Is life an electromagnetic phenomenon? In Bios Plasma, W/ed/Radekja Widonik. Kull, 1976, pages 73-81. Stewart G.T., Liquid Crystals in Biological Systems Molik. CRYST. 563-580, 1966. Varga A. Protein Biosynthese bei Mikroorganismen unter den Werken von Eiseren Elektromagnetisken. Feldern, Theme, Leipzig 1976. Weaver R. Elf Effects on Human Circadian Rhythms, in M.A. Persinger/ed/ Elf and VLF. Electromagnetic Field Effects. Plenum, New York, London, 1975, pages 101-144. Wojciechowski Z.W., W. Sedlak, J. Zahn, The Utility of Bioelectronics and Bioplasma Concept in the Study of Biological Terrain and its Equilibrium, First World Energy Medicine Congress, Paris, 18-20. November 1977, pages 127, 135. S. Manzarski, Physical Plasma in Biological Environment, Post P. Physiki, Tone XX, 3, p. 383, 1969, and Poznan, 1968, 3 Symposium on Radiospectroscopy and Quantum Electronics. Asound.c.d. Plasma Physics and High Temperature Chemistry. Plasma for Extraterrestrial Resource and Applied Technologies. Projet 93 pages, Plasma for Extraterrestrial Resources and Applied Technologies 27/02/1981. Reference Memorandum 3543. 81020 NASA Jet Propulsion Laboratory, Pasadena, USA. Asound.c.d. NASA Consultant in MEM 3543. 81020 October 30th-1981 Interim Report. Extraterrestrial Materials Processing, JPL, Pasadena, 148 pages. Asound.c.d. PhD Sciences Mention University Paris 7 and Degree 75779 BA 208 November 7, 1979. GF Values Determination Form NIL, 2356 A2158A, produced in Argon Plasma Ltd. Asound.c.d. Projet de Congrès de l'Académie des Sciences, CRAS, France. Les quantités d'origine biologique, base de information génétique. Mars 1982. Asound.c.d. Médecine quantique intronique c.d. Asound 1991 226 pages. Preface du Professor Dr. Helena Baranova. Geneticienne. Asound.c.d. Médecine quantique intronique et protection nucléaire pour notre biologie c.d. Asound 35 pages. Congress International Artic 21-22 May 2011. Asound.c. D. Proprieties radiatives de molecules et elements naturels cd. Asound, 5 pages. Origine d'une pathologie mémorielle dans les mécanismes d'immunodepression du HIV. People with News. 30.12.1994. Asound.c.d-HIV AIDS Congress, Metal Carbonyl Compounds, Preliminary Results in Cancers Associated to Immunotherapies. 12-13-14 Maggio 1995 Bocca di Magra, La Spezia, Italia, Grandi.mr.a. Canuto Maggiore. A. La Civita. Asound.c.dm.grandiem.maggioragi.numico r.canuto p. Corriere p. Sola Advances in Management of Malignancies iron carbonyl compounds preliminary results on breast and lung cancer December 6 to 10, 1993. Asound.c.d grandi.m canuto r.a corriere.p sola.p maggiora.m lachivita.a 16 International Cancer Congress 1994 New Delhi October 30 November 5. Rep 941030 P661 to 667 Glycan Industries LLC preliminary results for some responses in cancers with metal carbonyl compounds under associated immunotherapy. Metal FLNT4 complex, ZN-FAV others. Asound.c.d Resonance Serenkopf Flux Cosmic au niveau de cellules cérébrales Revue de Bio. Mathematiques P7423-7429 October 1968. Contact us at Glycan Industries LLC at glycangroup.com and Michael Glycan XR at glycangroup.com.